Welcome back, students, to DNA repair. Today, we are going to talk about a new type of repair mechanism called the post-replicative repair. You have previously come across the occurrence of thymine dimers and the different ways by which we could remove the thymine dimers. We saw about the photoreactivation system. We saw about the nucleotide base pair excision repair system. But sometimes what happens is when DNA polymerase 3 sees this thymine dimer, it will hesitate to go ahead. That is, it will leave certain gaps in between when it is replicating. And as a result, almost 800 to more base pairs, bases will be left behind. And in this strand, what happens? A long gap is being formed. And further on, the replication will be continued also. So at the region of the thymine dimer formation, you can see a thymine dimer here. Corresponding to that, DNA polymerase 3 gets confused and it leaves behind a large gap. Now, since a part of this strand is absent, now see, this strand is absent, some of this portion is absent. At the same time, there is also a thymine dimer over here. There appears to be a no viable template for replicating a new DNA. And unless and until we correct this, probably this would lead into a highly damaged DNA in the coming cycles. So, E. coli needs to correct it. So how can it do it? That's what we are going to learn in the sessions, in this session. Now, E. coli encounters such a situation of having thymine dimer along with the corresponding gap. If that occurs, uh, two strategies can be adopted. One is it can either use a polymerase which can replicate even these lesions. That is, even these lesions can be further replicated. In the coming daughter strands also, the, the lesions can be replicated. Such polymerases can be used. Now in E. coli we have an example is polymerase 5 which can do that. And the action of polymerase 5 has been found to be in such a way that in the regions where the thymine dimers are being formed, adenine containing nucleotides will be added by DNA polymerase 5. In the East, we have two options like polymerase Rev3 bar 7 and RAD30. And these two will also do a function of adding nucleotides even at the region of lesions. It does not remain confused there. At the regions of lesions, that is, it will add certain nucleotides over here. But in the case of RAD30, sometimes it might be error prone. So that one strategy to repair such mutated DNA containing thymine dimers as well as gaps is to use polymerases which can replicate their lesions. And another option that we have is the second one, which is depicted here. Okay, now regarding this, I need to tell you something. If you go to look at the replication fork, the replication fork has mainly what? It has two DNA duplexes. That is, one is here and another one is here. We can see that the strand D is complementary to the strand A. And so, if you want to replace this region of C, you can take some portion of the region corresponding to this gap from the strand A. And that would result in the repair of the particular gap which has been created by this thymine dimer. And that repair is being done with the help of a protein called the REC-A protein. In fact, it's a group of proteins which act here. And that group of proteins are referred to as a REC-A. And since this repair takes place at a gap created by the failure of DNA replication, the process is also called as a post-replicative repair. So, what is post-replicative repair? Because the repair takes place, what happens? after the process of replication, there is a failure of DNA synthesis here. That is, DNA replication has been done, and it's after that you are trying to repair it. And since the gap is created because of the failure of DNA replication, a repair mechanism, that repair mechanism is called the post-replicative repair. Let's go into the details of that. Since the Rec-A protein is being involved in this process, it is also known as a Rec-A dependent post-replicative DNA repair. Now you can see, look into here, you have a replication fork, one with a newly formed DNA strand, a fully properly formed DNA strand, but at the same time you have a strand over here which contains a thymine dimer, and here DNA polymerase is not able to synthesize these nucleotide bases, so it is skipping the thymine dimer during the DNA replication. DNA replication is not happening over here because the thymine dimer is being present over here. So, as a remedy to it, during post-replicative DNA repair, the protein Rec-A will come and bind to the blue strand over here, which contains the thymine dimer. And what it does, now this is trying to get information from the complementary strand which is being present here. So what will it do? It comes and base pairs with a complementary strand. With the help of Rec-A, the single stranded with the thymine dimer invades the normal sister duplex here. I think you got an idea. This one, which contains a thymine dimer, with the help of Rec-A, it is moving from here and it is coming towards this strand. This is a duplex, fully formed duplex strand that will become separated, and the blue strand is invading over here. That is, it is trying to take information from the fully formed 5' strand over here. Okay, so it's trying to swap some DNA or some information onto that. So once it is base paired with the fully formed 5' to 3' and the black one, okay, once the defective DNA portion is being attached to the what? It is being found attached to the black one. You can see the blue one is being attached to the black one over here. After that, what happens? Some endonucleases will come and produce two nicks on both the sides. Okay. See here as well as here, some nicks are being provided. And once the nicks are being produced, this black portion which is remaining attached to the duplex of thymine dimer will be transferred towards the defective DNA here. And a gap comes in the parent strand over here, which was properly replicated in the beginning. So you can look here, the black strand is absent over here, and that has been transferred towards the strand which contains the thymine dimer. So you're swapping or exchanging a particular strand of DNA from the newly formed properly synthesized DNA to the improperly synthesized DNA, complementary to the improperly formed DNA strand which had a thymine dimer. Thereafter, what happens? DNA polymerase 1 and DNA ligase comes and acts. What will happen? You can see here DNA polymerase action, and as a result of the DNA polymerase action, new DNA synthesis will happen, and the nicks which happen to be here would be sealed with the help of DNA ligase. And even now you can see that the thymine dimer still remains over here. Okay, but the gaps which were being found on either side of the thymine dimers are being removed with the help of the Rec-A dependent post-replicative DNA repair system. Thereafter, the thymine dimers will be removed by using various other enzymes as we had described earlier. So I think this is clear to you. And since the Rec-A locus is originally discovered and named in the recombination process, this post-replicative error is also post-replicative repair system is also known as the recombinational repair, and it shares many enzymes with the normal recombination process. So this process has got two names, known as a recombination repair system as well as a post-replicative repair system. I'll just show you a simplified version of what we had described earlier. So you have a replicator fork with a thymine dimer, and there is a gap over here, and you have a fully formed proper DNA in the A section. And so what happens here? A section of the A DNA is being swapped over to this particular region. It's the same thing. And thereafter, the DNA polymerase 1 and DNA ligase will act, and you will get a fully formed repaired gap. The gap will get repaired over here, as well as here the DNA from the sequence A is being swapped over, and you fill the gaps over here. The thymine dimer still remains, which will be further repaired by using certain other enzymes. I hope this session is clear to you, and we will be continuing on the various other DNA repair mechanisms in the coming sessions. Thank you for now.